cell, so we have a gradient to facilitate that movement. All right. So let's move on to speak about active transport. We already talked about some of the separating or unique features of active transport. It is non-spontaneous. It requires cell energy. That energy is usually going to be in the form of ATP, which is our currency, so to speak. It involves a pump of some type, and this is a membrane protein. So this is also a protein. So this is where students sometimes get confused. We had passive mechanisms that required protein, and we have active mechanisms that require protein. So you really want to differentiate what the gradient looks like and whether or not this process requires energy, okay? Here, the movement is uphill, so again, this requires some sort of input of energy to facilitate this movement. Let's look at this image here. So we can see our membrane separating the two sides of this cell here, but there are equal concentration of particles, of solutes or particles on either side of the membrane, and so we don't have a gradient, okay? We don't have a gradient. That's what really sets this apart from the other mechanisms we talked about. And what we see is that there's now movement happening through this, protein, right? This protein that's using energy, it's harnessing energy. We're going to talk about how it harnesses energy, and it's facilitating or pumping actively those particles from one side to the other until we now end up in steady state where particles are moving freely from one side to the other, but we have a net flux or a net accumulation of particles on this side, okay? Now, should that happen if we don't have a concentration gradient? No, right? Nothing should be moving if we don't have a concentration gradient. But because of this protein, because of this active pump, we use energy or harness energy, and we actively pump molecules from one side to the other, which would not ordinarily take place because there is no concentration gradient. Okay. So let's look at the characteristics of a pump. So this is a type of membrane protein. It functions as an enzyme. So it being an enzyme, what is it going to do to this reaction? Speed it up, right? It's going to go faster. It's going to be catalyzed because this pump is acting as an enzyme. And in such a way, we'll see that this requires energy to make this happen. So it's going to harness energy by cleaving this phosphate. We talked about ATP. We talked about the energy that was harnessed in that third phosphate. And so we can lyse that third phosphate, free some energy that can facilitate the movement or the function of this pump. Okay. This also has specific binding sites. So what we'll see, this is an example of sodium ATPase, which is an active sodium and potassium pump. And so it has binding sites for sodium here on the left, and then it has two binding sites for potassium, which we'll see on the right. And what this pump does is it actively pumps 3 sodium out of the cell and 2 potassium into the cell. This is a very, 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 very important mechanism. We're going to see this a lot throughout the remainder of this course, and even in the next, in ANP2. This is important because what it does is it sets up the ionic environments on either side of the membrane, right? Without this pump actively pumping these ions into the extracellular and intracellular environment, we would dissipate our gradient. We would not have those relative concentration of ions on either side of the cell. We would not be able to excite a cell. We would have no such thing as the resting membrane potential. And so this pump is very important to the function of a cell, okay? We'll talk about it more, so just bear it in mind. This is the sodium-potassium ATPase pump. What we see here is that pumps can also exhibit saturation simply because they have these binding sites. So we have sodium here, or 3 binding sites for sodium on the left, and so automatically once we've bound 3 sodiums, we're maxed out. Okay, we're maxed out. So this can be saturated, and so that's that leveling off that we can also see with active transport. Now, there are 2 types of active transport. There's primary active transport, there's secondary active transport, and we'll just distinguish what those, the 2 differences are. So primary active transport uses energy from the hydrolysis of ATP, okay? So as we said before, this type of movement, this type of transport requires some type of energy. And this is where the difference between these two types of active transport lies. Primary active transport is going to use energy from ATP. Right? Secondary active transport is not. We'll talk about what that is. So basically we have our pump again as the example. We have our sodium binding spots. We have this ATP molecule being associated with this enzyme, with this protein in the membrane. And we have the movement of 3 sodiums out of the cell and 2 potassiums into the cell. Okay? But the main take-home message is that we're using ATP, 1 molecule of ATP, to harness the energy for this transport. Right. Secondary active transport, we're actually using the movement of another molecule down its concentration gradient, and we're sort of stealing or piggybacking on that process, using that energy and driving another molecule against its concentration gradient. I'll say that again. So we're using one molecule, it's traveling down its concentration gradient, right? So there's a natural concentration gradient, there's more sodium outside of the cell. And what do you think is making that gradient? We just talked about it. This sodium-potassium pump, right? This sodium-potassium pump is setting up these gradients. There's more sodium outside the cell than there is inside the cell, because that pump is pumping or kicking 3 sodiums out, kicking 2 potassiums in. So what we have is a gradient for sodium naturally where it will go down its concentration gradient right back into that cell. And so when it moves into the cell, because this is downhill, because this is spontaneous, this actually releases some energy which another molecule can use or harness or piggyback on and go against its concentration gradient, okay? I hope that makes sense. We're basically taking the energy that's released as another molecule goes down its concentration gradient, using that energy to facilitate another ion in the case, or another molecule in the case glucose, that is moving against its concentration gradient. All right, so the energy that is harnessed from that movement drives the pump, which eventually moves the other molecule against its concentration gradient. Right? I'm going to say that 100 times because it sounds a little bit confusing at first, but hopefully it makes some sense as we look at a few examples here. So the first example is the sodium-linked glucose pump. Sodium-linked glucose pump. And because the movement is happening in the same direction, we sometimes call this co-transport. Co-transport, meaning that they're both sort of going in the same direction. This is an example of secondary active transport. We have the movement of sodium down its concentration gradient, and that facilitates this pump, which uses this energy to move glucose against its concentration gradient. Okay. Another example here is the sodium-linked proton pump. Okay. And so again, we're using the natural gradient that sodium has. Sodium is moving down its gradient. We're freeing up some energy, and that energy is being used to drive protons or to drive hydrogen against its concentration gradient, which happens to be out of the cell. Okay? And now in this example, the sodium-linked proton pump, because the movement is happening in 2 different directions, we sometimes call this counter-transport. Okay, counter-transport. So don't get too confused with the terminology. Co-transport, counter-transport, they're all forms of secondary active transport. Secondary meaning that we're not using ATP here, right? But we're using or harnessing the energy as another molecule moves down its gradient. Okay, and now this table here is a good way for you to compare these various mechanisms to test yourself. Make sure that you can come up with different scenarios, make up your own questions. I usually advise students to sort of block out some of these columns, block them out, and try reproducing that on your own. And not reproducing it from memory, right? Not memorizing this and then just regurgitating it, but rather making sure that you can fill in these blocks from understanding the concept, from understanding these differences. Okay, and then we have some examples down here as well, such as the sodium-potassium pump, some of the molecules that are being moved, right? We talked about these types of molecules. These move freely through the membrane. Fatty acids, steroid hormones, oxygen, carbon dioxide, et cetera, et cetera. Right, so I advise you to try to study and practice with that table. Okay, now I was sort of alluding to this idea a little bit earlier. We talked about the various ICF and ECF concentration of certain ions. Now we know typically at rest, a cell is more negative on the inside, more positive on the outside. We also know that the various ion concentrations are such that there's more sodium outside and there's more potassium inside the cell. And we know now that these relative concentrations are being achieved and maintained by the presence of some of these transporters that we talked about today. One being the sodium-potassium pump, and the other being these leak channels. These are leak ion channels that simply allow the movement of ions to go through them. And so these two mechanisms, this being a passive mechanism, this being an active mechanism, together they help to maintain those relative ion concentrations. We have the sodium-potassium pump on the one side. It's pumping sodium out of the cell, pumping potassium into the cell. And on the other side, these ions are leaking right in the opposite direction. And so together, these two forces, right, these two mechanisms are really establishing and maintaining those relative ion concentrations outside the cell and inside the cell, right? And just think about it. If we lost this pump, we would stop accumulating potassium inside the cell. We would stop accumulating excess sodium outside the cell. So these two pumps, right, this sodium-potassium pump, as well as the ion leak channels on this side, are very important for maintaining these relative ion concentrations, which we'll see later on. This is how we can excite a cell, neurons, okay? So very, very important function in our cell membrane.